Welcome everyone to the 9th webinar of the Science of Aging Series 2021, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. For this series, we've lined up a number of webinars, all focused on the science of aging, being conducted by leading researchers around the world in their relevant fields. This is Sarah McFarland from Inside Scientific, and I'm very pleased to be your host for today's event. Today's webinar is titled Cardiac Inflammation and Repair Following Myocardial Infarction, and will feature Dr. Mary Lindsey, Chair of Cellular and Integrative Physiology and the founding director of the Center for Heart and Vascular Research at the University of Nebraska Medical Center. In her presentation, Mary will discuss her research involving the physiology of recovery from cardiac events. Before we get started, we'd just like to acknowledge our partners at the APS and the Alliance for Aging Research, and also to thank the sponsors for this webinar series for making it all possible. And now our session sponsor would like to share a brief message.
Harvard Bioscience has been working with researchers and helping to advance life sciences for over 100 years. Our solutions are used around the globe in aging-related research, such as cardiovascular disease, diabetes, neurodegenerative disorders, motor and sensory disorders, infectious disease research, and more. From molecular analysis and whole cell or slice electrophysiology assays, to characterizing changes to organs or tissues in a controlled environment, all the way to whole animal stress-free physiological data collection and automated behavioral assessments. Our proven solutions, combined with world-class services and support, are used by the scientific community throughout many stages of their research, where we have been cited in over 375,000 research publications. Our team is with you every step of the way. Whether it's your first study or you've been working with us for years, let Harvard Bioscience be your partner in research.
And so, without any further delay, I'm very pleased to welcome Dr. Mary Lindsey to the floor. Mary, take it away whenever you're ready.
Hello, everyone. A myocardial infarction occurs when a coronary artery is occluded, shutting off the oxygen supply to the tissue downstream of the occlusion site. When this occlusion lasts longer than about 30 minutes, myocardial infarction ensues. In the mouse model, we generate MI by using a suture to tie off the coronary artery. Shown in the EKG at the top, a normal pre-MI EKG looks like this, and below, immediately after the MI surgery, you see ST segment elevation, indicating an ST segment elevated MI. By echocardiography, a normal time zero heart will show an elongated left ventricle, whereas by day 7 after the MI, the shape becomes more oval. In the top panel, TTC staining in a normal myocardium will show a strong red staining, whereas after the infarct by day 7, you'll see strong white staining or lack of staining where the myocardial infarction occurs. In response to the MI, the myocardium undergoes a time course of changes that initiate with cardiomyocyte death, which stimulates leukocyte infiltration and activation, and extracellular matrix degradation. This initial inflammatory phase initiates the repair process. This transcends into a wound healing phase that is represented by activation of the myofibroblasts and synthesis of new extracellular matrix to form a scar. This scar formation and the alveolar remodeling process culminate the response to MI. Our lab is particularly interested in the balance that occurs in the extracellular matrix between the destruction of the existing ECM and the construction of new ECM to form an infarct scar. On the left side of the equation, the matrix metalloproteinases are a family of proteolytic enzymes that govern the breakdown of existing extracellular matrix, which is needed to remove necrotic myocytes. When this side of the equation is overambitious, this can lead to excessive wall thinning in the infarct region, which can generate LV aneurysms, and particularly in the mouse model, permanent occlusion can end in rupture for approximately a third of the male mice. On the right side of the equation is the construction of new extracellular matrix that's governed by the fibroblast. The fibroblasts synthesize a number of ECM components that are necessary to generate the infarct scar. When the right side of the equation is overambitious, this can lead to a fibrotic left ventricle, which is a conduit for congestive heart failure and also is a stimulus for arrhythmias. The last thing to say about this slide is right in the middle sits inflammation. The macrophage and the neutrophil in particular serve roles on both sides. When they are pro-inflammatory M1 or N1 cells, they contribute MMPs to break down ECM. When the macrophage and the neutrophil are anti-inflammatory M2 or N2 cells, they contribute growth factors to stimulate the fibroblasts to produce more ECM. Our lab has spent a number of years developing signaling networks for the different cell types that are involved in the MI process. The first one shown here is the MI macrophage signaling network. At day one, the post-MI monocyte infiltrates into the infarct region to become a macrophage. It responds to a number of pro-inflammatory stimuli and also has a strong pro-inflammatory output, particularly IL-1 beta. Its primary cell physiology role is in promoting inflammation and promoting the breakdown of the extracellular matrix. By day three, the MI macrophage shifts to become a phagocytic cell, also becomes proliferative and undergoes metabolic reprogramming. By day seven, the macrophage is participating in the synthesis and deposition of extracellular matrix. In particular, macrophages at day seven can produce collagen in addition to a number of other reparative factors that are extracellular in nature. And so over the time course of MI, over the first week, the macrophage shifts from a pro-inflammatory to a reparative cell type. Likewise, the neutrophil also transitions over the first 7 days of MI. The day one neutrophil that infiltrates in from the blood also is a strongly pro-inflammatory cell. It has infiltration into the infarct as a primary role, but in addition, it is strongly pro-inflammatory and breaks down ECM by secreting a number of MMPs, including MMPs 8 and MMP 9. By day three, the neutrophil shifts to undergoing an apoptotic reaction, and also those neutrophils that are still present use fibronectin signaling. Cathepsin proteases are also activated at day three in part to stimulate the apoptosis. But then there's also the beginning of initiation of ECM reorganization that transitions to day 5, where MMPs are now inhibited in the neutrophil by having much lower expression. And also ECM deposition is promoted by the neutrophil as it converts to an N2 anti-inflammatory and reparative neutrophil. And then this phenotype persists to day 7, where again, ECM synthesis and scar formation are predominant roles for the day 7 neutrophil. And finally, the fibroblasts after over the MI time course shows a continuum of phenotypic changes. The day 0 fibroblast is a homeostatic cell there to maintain the normal physiology of the left ventricle. At day 1, the fibroblast converts to an inflammatory cell type that also prevents its migration away from the infarct region. At day 3, the MI fibroblast undergoes a proliferative upregulation and also becomes proangiogenesis and profibrosis. and begins secreting ECM components. This ECM production persists to day 7, where the fibroblast is the primary contributor of the scar that gets formed. At this time, though, anti-angiogenesis is stimulated in the fibroblast, and it shifts from a pro-angiogenic phenotype at day 3 to an anti-angiogenic phenotype at day 7 to shut off the signal to form new vessels. By day 28, the infarct scar is formed, and the fibroblast will persist throughout the history of the infarct, serving as the primary way to maintain that infarct scar. And so throughout the lifespan of the infarct, you will continue to have cardiac fibroblasts, and they will be reduced in number and activity, but they are still there to maintain that neo-homeostasis that is formed as a result of the MI. And so I want to show you a vignette of a recent study that we've done looking at the role of exogenous IL-4 to modulate the neutrophil in the macrophage. And so IL-4 is lowly expressed after MI, but in both the macrophage and the fibroblast, the receptor, IL-4 receptor, is strongly present at all times post-MI, indicating that macrophages and fibroblasts can respond to IL-4 if it is presented. And so using that background, we hypothesized that an exogenous IL-4 infusion started 24 hours after MI would promote a polarization switch in the inflammatory cell phenotypes. To address this hypothesis, at time 0, we performed echocardiography to establish a baseline echo and then gave the MI surgery. 24 hours later at day 1, we did echo again to confirm infarct and to confirm that that echo is similar between the two groups before treatment was started. And then we started IL-4 infusion by mini osmotic pump from Alzet, and we infused IL-4 until day 3. At day 3, we performed echocardiography and then sacrificed some mice. And I should back up and say that we also gave saline as the control untreated group. And so at day 3, when we sacrificed, we isolated neutrophils and macrophages from the infarct, which included the border region as well. They were all taken as one unit. And so we looked at a number of pro-inflammatory markers at day 3 post-MI. And when IL-4 was given in vivo and then the neutrophil was examined ex vivo, there was a reduction in a number of the pro-inflammatory genes. CCL3 was reduced, IL-12A was reduced, TGF-beta1 was reduced, and TNF-alpha was reduced. There was a couple of cytokines that were not changed, and IL-1 beta in particular indicates that because the treatment was started at 24 hours post-MI, it makes sense that IL-1 beta was not changed because it peaks at that time point. None of the anti-inflammatory markers that we looked at arginase 1, IL-10, MRC1, or YM1, none of those were affected with IL-4. We also looked at tissue clearing at day 3, and saline and IL-4 had very similar values of tissue cleared in an ex vivo assay. And this is again consistent with infarct sizes being equal because the treatment was started at 24 hours after MI. And it's also consistent with us not affecting that initial pro-inflammatory phase. We didn't start changing anything until 24 hours after MI. In contrast, the macrophages, rather than showing a decrease in pro, they showed an increase in anti-inflammatory markers at day 3 post-MI. So compared to the saline, the IL-4 had elevated arginase 1 and elevated YM1, and those were the only two genes that were elevated in the day 3 macrophages. None of the pro-inflammatory markers changed, and these two anti-inflammatory were also not changed. And so this indicates that the predominant effect of IL-4 was to promote the conversion to M2 macrophages. So to evaluate the cell physiology of the macrophage at the day 3 post-MI, we isolated out macrophages from the infarct border zone and performed ex vivo staining to evaluate their in vivo phagocytosis. And so what we did here is we have saline on the left and IL-4 on the right. In the top panel, we have stained the macrophages for neutrophil component, Ly6B2, using FITC conjugated antibody. And what this shows is the macrophages that have neutrophil components taken up inside of them. The bottom panel is for alpha-actinin, showing the myocyte components that are within the macrophage. And what we see in the top panel with the neutrophils is IL-4 treatment stimulated the macrophages to uptake more neutrophil components than it did in the saline controls. Whereas in the myocyte uptake was not different in the macrophages, which again goes back to the fact that the infarct sizes were equal between the two groups, and the treatment was started 24 hours after MI, and so that allowed all of the initial necrosis and inflammation to proceed, and then we started the treatment. And so this indicates that phagocytosis of neutrophils is upregulated with IL-4 treatment. And so then we performed RNA sequencing to evaluate the macrophage gene expression patterns in the saline and IL-4 treated groups. And the first thing we did was to assess the purity of the macrophages that we had isolated. And so we looked at a number of genes that are macrophage specific and showed expression of those genes. In contrast, endothelial cell, fibroblast, lymphocyte, myocyte, and neutrophils all had negative gene expression in the isolated macrophage pool, indicating that this was a pure isolation of the macrophages. Of interest, CD14 was reduced in the IL-4 treated group, and CD68 and FCGR3 were both elevated in the IL-4 treated groups. And so looking at the proliferation genes that were expressed at day 3 post-MI in the macrophage, none of these proliferation genes were changed between groups. TUB-A1 was the only gene that was elevated with IL-4 treatment, indicating that IL-4 can stimulate proliferation of the macrophage through this mechanism. And so looking at the ex vivo macrophage gene expression of known phagocytosis genes, we evaluated these 8 genes. Of these 8, FCGR2B, MERTK, and MRC1 were all elevated when IL-4 was used to in vivo treat the macrophages. And so these 3 components match up with the neutrophil phenotype and indicate that the increase in the neutrophil phagocytosis is likely due to the contribution of these specific genes. We also evaluated macrophage phagocytosis in vitro using peritoneal macrophages and stimulating them with IL-4. When we did that, the 3 components that we had shown ex vivo were also elevated with IL-4 treatment. In addition, there were 3 other genes. 2 of them were decreased and 1 of them increased with the in vitro IL-4 treatment. And so again, this just highlights that the in vitro conditions often don't mimic what you see ex vivo or in vivo. And so every time you think about in vitro, you should also think about having a companion in vivo piece. And so by principal component analysis, we see that IL-4 treatment shifts the macrophage polarization profiles such that they are distinct from the saline controls. By important feature analysis, PDGFC comes up as the most highly upregulated factor in the IL-4 treated macrophages. And this is just a volcano plot showing the same data visualized a different way, showing that PDGFC has the highest p-value change and is also highly elevated by fold change. In Go term analyses, the top upregulated features in the day 3 IL-4 treated macrophage are shown here, and the top down-regulated processes are shown here. And so in summary, IL-4 treatment in vivo has distinct effects on the neutrophil and the macrophage. The neutrophil has a downregulation of a number of pro-inflammatory genes to reduce its N1 activity. And the macrophage, in contrast, has increase in arginase 1 and YM1 to increase its M2 activity. The macrophage also has an increase in phagocytosis that is distinguished by upregulation of a specific gene expression pattern. On the right side of the slide, the reduction in the PDGF receptor alpha is linked with a stimulation of an increase in PDGFC that is seen in the IL-4 treated macrophages. There's also a number of MMPs, collagens, and TIMPs that are either downregulated or upregulated to shift the pattern of ECM deposition and breakdown that is observed after MI. And so all combined, these factors work together to increase inflammation resolution and accelerate the transition to the reparative phase when IL-4 is presented exogenously. And so I want to shift gears for a minute and talk about some of the matrix metalloproteinase work that we've done over a number of years. And a few years ago, we developed the cardiac metalloproteinase actions, the karma postulates, to assign physiological roles for different MMPs in the setting of MI. And so the first postulate is that the MMP changes in all cases of MI. The second postulate is that MMP actions can be mimicked in vitro. And the third postulate is that modulation of the MMP alters cardiac remodeling. And then finally, the fourth postulate is that the MMP proteolytic product, the substrate, likewise regulates cardiac remodeling in a parallel way that assigns MMP and substrate in the same pathway. And so there is a number of evidence shown here on the right that is from research over the last 25 years, and not just in my group, but in many labs where we have all contributed together to better understand how MMPs serve roles in the remodeling. And so we now know the expression pattern of MMPs after MI. We know that different MMPs are expressed in different cell types, and then they have different functions after that. We also know a lot of studies looking at either global MMP inhibitors or specific MMP inhibitors, as well as null and transgenic animal models for particular MMPs, but then also their tissue inhibitors of metalloproteinase, their TIMPs. We know those effects on post-MI remodeling. And then finally, there have been a few studies, in particular, for example, by Dr. Alessandro de Castro-Bras at East Carolina University, looking at the roles of the MMP substrate and what effect the cleavage of that substrate has to regulate LV remodeling. And so our lab in particular has contributed the MMP9 Karma Knowledge Map, where for MI, we know the inflammatory mediators that are produced, in particular the cytokines, chemokines, growth factors, and extracellular matrix components that stimulate the macrophage and the neutrophil to signal through particular chemokine and cytokine receptors, as well as integrins and a number of other signaling pathways to stimulate the secretion of pro-MMP9. When pro-MMP9 is secreted, we know ex vivo, in vivo, and in vitro, what activates the pro-MMP9 to become active MMP9. And then once it's activated, we know what inhibits active MMP9. For example, alpha-2 macroglobulin and all four of the TIMPs. And then when MMP9 is activated and it proteolyzes substrates, we know which substrates are relevant to the MI setting. And we know what effect their cleavage has on different cell types, in particular for the leukocytes and the fibroblasts, how these substrates can affect the polarization profiles of these cell types. And for the macrophages, how it regulates phagocytosis, and for the neutrophil, how it regulates apoptosis. And then from the cell effects, we also see global left ventricle effects on scar quality and quantity, as well as the generation of left ventricular dilation and impaired physiology that occurs after MI. And so over the last 25 years, we've accumulated a lot of knowledge regarding the role of MMP9 in the setting of MI. Our lab has also recently undertaken some studies looking at MMP12, a different MMP, and how it affects MI remodeling. And so MMP12 was originally called macrophage metalloelastase. And so we know that MMP12 is highly expressed in macrophages. In a study performed by Dr. Rahmani Iyer, she showed that after MI, MMP12 protein, but then also RNA, is highly elevated immediately after MI, even at day one. We see it go up and stay up through the first 7 days of MI. And Dr. Iyer showed that if when she isolated neutrophils from no MI blood and then isolated them from the day one LV infarct region, that there was an upregulation of MMP12 RNA shown in those neutrophils. And so in addition to the macrophage, the neutrophil is also a novel source of MMP12 in the MI setting. And so Dr. Iyer showed that if MMP12 is inhibited using this specific MMP12 inhibitor started 3 hours after MI, and then the hearts were evaluated at day 7, she showed that it worsens LV geometry and physiology. So at time 0 before the MI, if you compare the wall thickness of the infarct region, there is wall thinning that occurs over the first 7 days of MI. The LV remodeling index goes up, volumes go up, ejection fraction goes down. This is a typical MI response at day 7 in the mouse permanent occlusion model. With the MMP12 inhibitor, those numbers got even worse. So the wall thinning was even more pronounced. The remodeling index was higher, volumes were higher, and EF was even lower after MI. And Rahmani showed that this was due to increased late ECM degradation. I told you earlier that the neutrophils come in at day 1 and produce proteases that break down ECM, but then by days 3, 5, and 7, the neutrophils should be undergoing apoptosis and or converting to a reparative phenotype, such that there's very little ECM degradation after day 3. What Rahmani showed was that this increase in late ECM degradation was due to the prolonged inflammatory status of the neutrophils, that they were staying around and staying a pro-inflammatory neutrophil at day 7. And so she also showed that MMP12 regulates neutrophil apoptosis, that the MMP12 inhibitor reduced CD44, it reduced hyaluronic acid degradation, all which serve to reduce caspase-3 cleavage and to reduce neutrophil apoptosis. She then did the transverse experiment and in vitro stimulated neutrophils with MMP12 and showed that it had increased apoptosis measured by cleaved caspase-3. And so this showed that MMP12 was a direct stimulator of neutrophil apoptosis. And so these results suggest that there's a protective role for MMP12 in the MI setting. And so a couple of take homes here is that every MMP has some common features with other MMPs, but also some distinct novel characteristics that set it aside from the other MMPs. And then the second component is that even though something goes up, it doesn't mean that inhibition is the optimal strategy for therapeutic options, and that sometimes the factors are going up after MI as a way to serve a cardioprotectant role and try to be a resolution promoting factor. And we believe that MMP12 is one example of a resolution promoting factor where it's actually trying to do benefit after MI. And so for us, MMP activity is the beginning of the situation and not the end of the study. And so all of our papers that look at MMPs, we start figure 1 with showing that the MMP is elevated and then going from there to show what role it serves in the heart after MI. And that has proved to be a pretty effective strategy in us finding some novel roles for the MMPs after MI. And so in conclusion, myocardial infarction is a predominant cause of congestive heart failure. And by identifying the relationships between inflammation and fibrosis that stimulate adverse remodeling, this provides us with mechanistic insight into what is occurring after MI in the left ventricle. And it also helps us to identify those targets that will help us to know the patients who are at risk for LV remodeling that will progress to heart failure, and that if we can develop early aggressive therapies to help those patients in particular, then this might serve a beneficial strategy for limiting remodeling. On the right side is a word cloud where we took the titles of all the papers we published, and every time a word is used in a title, the word gets larger. And so this just demonstrates that our lab is really focused on matrix metalloproteinases and their roles in myocardial infarction. And with that, I will thank you and stop to take questions.
All right, great. Thank you for that awesome presentation, Dr. Lindsey, and we will move into our Q&A now. So let's bring your camera on. There you are. Okay, so very first question here is infarct not confirmed at the time of ligation of the anterior descending coronary by visual inspection or ischemic blanching?
That's a great question. Yes, we do confirm MI at the time of surgery. We also do EKG monitoring. So during the entire surgery, we're monitoring EKG and can confirm ST segment elevation at the time of MI. We do the echo at day 1 before we start the treatment, just as another confirmation in terms of the rigor and reproducibility of the study, just, double downing that we know we have infarct. We know they're similar between groups. We're not introducing any bias because of that. So to answer your question, yes, at the time of surgery, we do confirm by EKG and by visual blanching.
Great. Okay. Someone has also asked, have you noticed any difference in these results depending on the sex of the mice?
Yeah, this is a great question. There's still lots to be done looking at potential differences in between sexes in terms of MI remodeling. I will say that Christine de Leon-Pinel, when she was with our group, did a very nice study showing that the neutrophils that come into the heart after MI in the female, there are lower numbers of neutrophils than in the male mice. And at the same time, the tissue clearance rate is equal between them, which indicates that the neutrophils from the females are more efficient at clearing tissue than the male neutrophils. So that's just one example. There's a lot of similarities. There are differences. This is an area wide open for exploration. And this is just a good point here to plug AJP Heart, which has a recent call for articles that consider sex as a biological variable. And so we're highly interested in more folks in the community looking at both sex differences, but also sex similarities in MI remodeling.
Awesome. A really cool factor to explore, I think. All right, the next question here is, does IL-4 alter checkpoint signaling? Because this seems to be important for macrophage activity under stress.
Yeah, that's a great question. Now that you've asked that, Jeff, I'll have to go back and look at that data set. I do know that the PDGF receptor alpha has a connection with cell transition and with checkpoint signaling. So the answer is likely yes, and we have that data, so we'll probably be able to know, the specific answer to that question. But we didn't look at that when we were writing up that study.
Okay. The next question here is, does IL-4 treatment have an effect on the cardiomyocyte, for example, hypertrophy or atrophy pathways?
Yeah, and that's a good question. So in the IL-4 study, the only part of the myocyte component that we evaluated, besides infarct size, which wasn't different because of the late treatment start time, the only thing of the myocyte that we looked at was the uptake of necrotic myocytes into the macrophage using phagocytosis. And in that case, there was no different between the groups, but that's more of a reflection of the macrophage physiology, I think, than the myocyte. We did not evaluate hypertrophy or atrophy, but I will say we didn't see any apparent differences that would suggest that those might be avenues that were, worth evaluating.
Okay. Great. Okay. So the next question here, well, first it's a compliment. Great talk, Dr. Lindsey. Then the actual question part is, are the N2 neutrophils observed at day 5 to 7 a newly recruited population of neutrophils, since many of the first wave neutrophils undergo apoptosis?
Right. This is a good question to ask. And so we don't have direct evidence to say concretely that where the source is, but I will tell you the indirect evidence that we have. We think, like you think, that the neutrophils that come in the first wave and then undergo apoptosis, we think that those are distinct cells from the cells that then infiltrate later and convert to the N2 phenotype. Based on the studies that we've done, it looks like the inflammatory cells, whenever they come into the environment, whatever environment they come into, dictates what cell type they become. And if they polarize to the pro-inflammatory or the anti-inflammatory or somewhere along that spectrum, the environment at the time of entry seems to be the dictating consequence of what's going to happen. I'll also point out that Matthias Nahendorf and his group at Mass General have done a number of studies with the macrophage that indicate that for the macrophage that is true, that the cells that come in at day 3 are distinct from the cells that come in at day 1. And at day 3, they're more proliferative, for example, and they're converting back to that resident macrophage phenotype. And so there's definitely a lot of indirect evidence in the field to indicate that the day 5 to 7 neutrophils are a newly recruited population.
Fantastic. Great. Okay. And I just want to mention too that someone that you mentioned, Matthias, just presented in another webinar with us. So if you're interested in checking that out, definitely head to our website to find his presentation. So the next question here is, how does the MI environment change with aging?
Okay, yeah, this is a great question, and so it's from Dr. Lysandra de Castro-Bras, who probably could answer this question better than I can. She's done a lot of studies looking at aging effects on LV remodeling, and so my answer to that is to point everyone to her literature, and so there are changes in the MI environment with aging. I will say that in the number of studies we've done in mice, up through 18 months, up until the time that they would be considered old, there is still a lot of similarities, particularly in the male sex. At that point, though, you can see distinct differences in between young and old females, going back to the sex difference question that was asked earlier. I think that some of the sex differences come out more if you're looking at an aging cohort. And then after 23 months of age, when the mice are becoming senescent, then then you have baseline differences in the aging left ventricle that contribute to differences in how they respond to MI.
Fantastic. Okay. Our next question here is, do you measure MMP enzyme activity? And if so, what is your preferred method?
And so this is kind of a old school question because, back in the day in the, 1990s, when MMPs were first being measured in the heart, gelatin tomography was like the go-to way. And when I was doing my dissertation defense, that was how I measured MMP9 for pro and active forms. I think that currently there is not the need to measure, to quote, measure MMP activity as much as there was in the past. Now, I would say the more important index of MMP activity is the generation of substrate fragments. And so especially in vivo, if you want to document that an MMP is elevated and that it has an activity, the best way to do that is to show that it has a consequence. And so, for example, when we directly simulated neutrophils with MMP12, we didn't, we used an active recombinant, but, we didn't need to measure activity of the MMP12. What we measured was the effect of the activity, and that was on stimulation of the caspase pathway to induce apoptosis. And so in terms of the MMP field, there's not the reliance on the activity assays today that there were, 20 years ago. Right now, most of this is based on substrate analysis.
Okay. Fantastic. Great answer. So the next question here is, how are T cells involved in this remodeling process? Is there a shift to Th2 or Th17?
And so for this question, I am 100% going to direct you to Dr. Christine DeLeon-Pinel at the Medical University of South Carolina and the research that she's been doing over the last 3 years to answer exactly this, what the T cells are doing after MI. There's definitely a shift in T cell repertoire during this time phase of MI. In addition to Christy DeLeon-Pinel, Stefan Franz and the Wurzburg group has also done some, very nice studies looking at T cells in MI. It's definitely not something that our lab is investigating because there's so many, good labs that are. T cells are definitely lower, much lower in total number than, for example, the neutrophils or the macrophages. It doesn't mean they're not important. It just means that there's definitely some technical challenges to being able to look at T cells, particularly at the individual cell level, because of those volume issues.
Okay, great. We've got lots of questions here. Just as a reminder, if you do have questions for Mary, please send them in through that Ask a Question panel. So I'm going to continue on here. So this next question is there any evidence of exercise protection prior to acute myocardial infarction?
The answer is yes. That's not work that our lab does, but there's lots of people who exercise mice and rats and humans before and after MI. And there's definitely evidence that exercise is protective.
Okay, great. Next question here. This is a long one, so I'm going to read it. The inflammatory response in the heart appears to be similar to that in injured skeletal muscle. Skeletal muscle has resident stem cells, known as satellite cells, that enable myofibers to regenerate, which do not exist in the heart. Do you see a mechanism for incorporating stem cells in myocardial repair/regeneration?
Okay, Steve, I see you're asking this question. I'm not going to answer anything about repair and regeneration of cardiomyocytes in the heart. I think there's been enough literature put into the field that people can make conclusions about the promise of stem cells for MI. That's definitely not something we look at.
Okay. The next question here is, do you think IL-13 will have similar effects that like IL-4?
Yeah, that's a great question. It's definitely possible. I will tell you that IL-10, which is similar to, it's another anti-inflammatory cytokine. IL-10 has definitely have a lot of positive effects on MI remodeling. Our group showed benefit of IL-10, and then Raj Kishore at Temple has done a number of studies looking at IL-10. And so definitely there is some rationale for thinking about IL-13 as well. And by the way, that data set we have for the IL-4 macrophages, the RNA sequencing data set, if anyone wants that, that's part of the publication. It's a supplemental table. So you can feel free to see if IL-13 is changing in that setting, or if it gives you any help in rationalizing your experimental designs.
Absolutely. A great resource for people. Thank you for that. We've got lots more questions here, so I'm going to keep going if you're okay with that.
Sure.
Did you expect any difference in the inflammatory reaction caused by a permanent LAD ligation MI model versus an ischemia reperfusion MI model?
Well, so AJP Heart has just published a couple of articles that are really good in providing insight into this question. And so one of them was an editorial on the difference between permanent occlusion MI, non-reperfused MI, and then reperfused MI. And so definitely those are distinct pathological responses, both of them involving inflammation. But for example, reperfusion brings in more inflammation sooner. And so, definitely there would be the potential for, some differences in response between those two models. I will say that we use the permanent occlusion, the non-reperfused MI model, because we're interested in heart failure and the progression from MI to heart failure. And that is the clinical scenario that is best mimicked with the permanent occlusion model.
Okay. And the next question here is, any functional improvement with IL-4 treatment?
Yeah, that's a good question, because I did mention that we did echo. And so we evaluated these mice at day 3, which is a somewhat early time point in terms of looking at the full dilation response to MI. And so in summary of the results, echo was none of the echo variables that we measured were different between saline and IL-4 treated mice. We don't necessarily think that is a bad thing, because since the treatment was started at day 1 and then we sacrificed at day 3, this was a short window of therapy. And the fact that macrophages and neutrophils are changing, but the cardiac physiology didn't change, indicates that those changes in the cell types are occurring before physiology. And so this is indicating that, molecular changes can drive cell changes, and the cell changes can drive LV changes. Others have reported that IL-4 treatment given for longer periods, I think it was 14 days, did show a difference in cardiac physiology. And so our time point of day 3 was too early to see a difference.
Okay, that makes sense. Great. All right. So the next question here, she's asked, I was wondering, how would this macrophage realm change in the setting of MI in the presence of metabolic syndrome, where one might have pre-existing systemic inflammation already?
Right. That's a great question. And so I'll point you to Dr. Ganesh Halade in his work. He's at University of South Florida in Tampa, and he's done a number of studies showing the effect of aging plus high-fat diet plus DHA as a modifier, and then giving MI. And so definitely changing either aging or nutritional status or underlying inflammation, any of those components can trigger shifts in how the MI remodels.
All right. This next question here is, how does changes in cardiac inflammation post-MI contribute to changes in cardiac metabolism and vice versa?
So can you say that one again?
Yep. How does changes in cardiac inflammation post-MI contribute to changes in cardiac metabolism and vice versa?
Yeah, that's a good question. So there's a definite tie-in with metabolism, and the whole concept of immunometabolism is something that is just starting to get thought of in the cardiac community. And so again, Matthias Nahendorf, another plug for his talk, he's done a lot of looking at metabolic shifts in the macrophage over time with MI. And so, it all interrelates. If there's changes in metabolism of whatever cell type or the whole organ, then it's going to feed into changes in the inflammatory status.
Great. Yep. And as Mary mentioned, he does have a talk on this in our immunology series with the APS as well. So definitely head to our website to check that out. I think I'm going to make this our second last question just based on time. Can reperfusion after ischemia alter the time course changes in the inflammatory/reparative process?
The answer is definitely yes. And Nick Frangogiannis and Mark Entman and a number of folks who have studied reperfusion over the years, can tell you there's definitely changes in the kinetics of cell infiltration. There's differences in the numbers of cells that come in. There's differences in the transition from pro to anti to reparative phenotypes. So all of that can be modified when you add reperfusion into the equation.
Great. All right. And our last question here, which is one of my favorites. Where do you see this field heading in the next 10 years?
Yeah, this is a super exciting time to be in the MI remodeling field because, 25 years ago, we had a lot of questions that we didn't have the tools to answer. And right now, with RNA sequencing, with single cell sequencing, I'll also make a huge plug for proteomic. With proteomic technologies, what they are right now, we are just poised to answer so many of these questions that have been on the back burner for so long. And so I think over the next 10 years, we're just going to see an expansion of the information that we are provided to help us understand better the whole MI remodeling process. And, in our lab in particular, the focus where we're headed is trying to modify this system in a way that does benefit. And so we know if we have a global change in inflammation, that ends up being a negative contribution to MI remodeling, that if you have too much or too little inflammation, that's not good. And so you want right at that happy medium where wound repair can occur and scar formation can occur, but you don't get too little or too much, because then you have detriments on both sides of that equation. So getting that U-shaped curve to be optimized by tweaking the system is something that is really the next generation of thinking, is how do we tweak it rather than how do we totally inhibit or how do we totally promote, because I think that so much of this isn't a linear process, it's a U-shaped process.
Absolutely. It's a very exciting time. Okay, well, I think I'm going to finish off the Q&A there on that really awesome high note. I really just wanted to thank you so much again, Mary, for all your fantastic insights today. It was a pleasure to have you with us.
And thank you for having me. And I didn't answer all the questions or comments, but if you had something to say and I didn't get to you, feel free to reach out to me.